Good day, everyone. My name is Oladapo Olawale Afolabi from the Lab of Vascular Biology, UFMG, and the title of my presentation is The Impact of Severe Caloric Restriction on Cardiac and Vascular Remodeling. Caloric restriction involves a 10 to 40% or in severe cases up to 50% reduction in the daily average caloric intake without causing malnutrition and affecting the intake of essential nutrients like vitamins and minerals. While high-calorie diet increases cardiovascular disease risk factors such as dyslipidemia, hyperglycemia, and diabetes, calorie restriction, on the other hand, causes certain metabolic adaptations, as you can see on the screen, which leads to cardiovascular physiological adaptations that we can also see on the screen, which cumulatively has profound clinical outcomes like the reduction in the risk of coronary artery disease, reduction in the risk of stroke, reduced cardiac arrhythmias, reduced peripheral artery disease, and reduced heart disease. Caloric restriction is proposed to work either by stimulating SIRT or creatine kinase protein to activate endothelial nitric oxide synthase, which leads to the increased production of nitric oxide, which now consequently has profound effects on the cardiovascular system. With this information, we therefore hypothesize that severe calorie restriction in mouse will have cardioprotective effects in terms of its impact on cardiac and vascular remodeling following ischemia-provoking pharmacologic and surgical insults. We prove this by evaluating the mechanisms involved in vascular adaptation under caloric restriction, by evaluating the possible vasculoprotective effects of caloric restriction in a model of expansive arterial remodeling and new intima formation, and by evaluating the possible protective effects of caloric restriction in cardiac hypertrophy due to administration of isoproterenol. Methods involved in the analysis of our samples include real-time PCR, immunohistochemistry, immunofluorescence, histology, morphological evaluation, Western blot analysis, and vascular reactivity experiments. For the animal model, there is postpartum placement of the mother on 50% reduction in calorie intake, while after weaning, the pups themselves are placed on 50% reduction in calorie intake, and all this is achieved by pair feeding, after which the animals are sacrificed and samples are collected. Following calorie restriction in the young pups for 30 days, both pharmacologic and surgical treatments are employed. Pharmacologic treatment involves the administration of isoproterenol subcutaneously for 10 days at a dose of 3 to 10 milligrams per kg per day to induce cardiac hypertrophy, while carotid artery remodeling with new intima formation is induced by the permanent occlusion of the right common carotid artery for up to 14 days. Vascular function by vascular reactivity experiments using concentration response curves and ex vivo cardiac function using Langadorf apparatus will be evaluated. Preliminary results from vascular reactivity experiments show that caloric restriction causes a reduction in vascular contraction and improved vasodilation. And then we ask whether these results can show a protection in the rats against some hypertensive and atherogenic stimulus, and this will offer a great insight into the treatment of hypertension and atherosclerosis. Again, administration of isoproterenol induces cardiac hypertrophy. However, calorie restriction causes a reduced expression of hypertrophy markers such as MHY6, MHY7, muscle alpha-actin, while increasing the expression of cardioprotective biomarkers such as atrial natriuretic peptide. Similarly, we observe that calorie restriction has an effect on serum cytokines profile by increasing anti-inflammatory biomarkers and reducing pro-inflammatory biomarkers. And then we wonder whether this data could be related to a possible prevention of cardiovascular damage related to inflammation from endothelial dysfunction, cardiac hypertrophy, and new intima formation. In conclusion, preliminary results show that severe calorie restriction reduces vasoconstriction, cardiac hypertrophy markers, and systemic inflammatory cytokines, while conversely improving vasodilation, cardioprotective markers, and anti-inflammatory cytokines. All these put together show that severe calorie restriction promises profound beneficial impact on the cardiovascular system and could prevent or retard cardiac remodeling, and this has profound implications on hypertension management and cardiovascular damage. Finally, a very big thank you to my research group in the Lab of Vascular Biology and to CAPIS for providing the funds. Muito obrigado.